Hi, my name is Sophia Spivey, and my project is the identification of the TolQ region involved in the maturation of the P1 virion. I did my research in Dr. Larson's lab at Bowling Green State University. Let's dive right into the abstract. The plasmid PRA031, which carries the TolQ gene under the control of an arabinose-inducible promoter, will be mutagenized in vitro using hydroxylamine, which results in GC to AT transitions. Plasmids thus treated will be transformed into delta TolQ strain RA1027. The lack of the TolQ protein in this strain results in a loss of the outer membrane integrity, rendering RA1027 very susceptible to killing by the detergent deoxycholate. This provides the ability to select for plasmids that make a TolQ protein that makes normal associations with other tool system proteins, indicating that it is properly trafficked to and inserted in the cytoplasmic membrane. The detergent-resistant survivors will then be tested for resistance to killing by P1 virus. Plasmids that provide for both resistance to detergent and the P1 will encode TolQ proteins that contain the features needed for normal TolQ function, but lack the features needed for successful assembly of the P1 virion. The DNA sequences of the TolQ genes carried by these plasmids will be determined to identify putative TolQ sites that support the assembly of the maturation of P1 virions. Next, let's move on to the plasmids used. We used PRA013 and PRA014. As you can see, PRA013 has PBAD, TolQ, TolR, TolA, and term, whereas PR014 has PBAD and term. One important thing to note is that PRA014 was made from PRA013 by engineering a deletion using a PCR-based strategy. PRA013 is usually very plasmid sensitive, whereas PRA014 is not. So if you move on to the table shown, where it's labeled plasmid sensitivity, you can see that every time we used PRA013, it was very plasmid sensitive, positive results, a lot of sensitivity shown. Whereas every time we used PRA014, there was little, if any, sensitivity shown, but majority of the time there is no sensitivity shown. The only time we had sensitivity shown is when we used B. If you look a little bit to the right, there's a picture of when we used E3 to show plasmid sensitivity. So you can see that 1016-13 and 1035/13 are the only two on there that show any plasmid sensitivity. So every time PRA013 is present, it is sensitive. When we had the PRA014, no sensitivity at all. The next thing I want to talk about, and it's kind of the last thing that's shown on here, is we wanted to talk about P1's ability to create plaques. So we did dilutions to show how far we can go with plaques, to kind of do plaque counting, to see how much we had present in a little solution of P1. So if you look at the control, there are no plaques present, which is perfect. That's exactly what we want, because it shows that there aren't any plaques present, and there aren't supposed to be any plaques present in that because there are no P1 in there, meaning no contamination, which is exactly what any scientist wants to see when you do a experiment. And then when we look at the P1 dilution, we use 10 to the negative 1 as our example, because you can see that whole thing is just covered in beautiful plaques, which kind of leads into the next step that we want to dive into, which is really believing or knowing that further research is able to be done. And we have a good starting point with this because we do know that what we're working with is actually P1, and we have a nice amount present. In the end, I also just want to say a big thank you to the Center for Undergrad Research and Scholarship for the financial support because it really gave me an opportunity to get more involved in the lab, learn a lot, and hopefully use this as a stepping stone to get into my master's thesis. Thank you guys so much and have a great rest of your day.